I'm going to really just dive in and spend the first 10 minutes telling you some background information about why you should care about mechanics. And the lovely talk that we just had about mitotic spindles just underscores this. And then from there, I'm going to describe tools. Really, it's a very tool and method focused talk that I'm going to tell you today. So to start with, I don't think I need to say this for anybody here, but whether you look at this video of fibroblasts or you look at this video of a T cell attacking a cancer cell, it's very clear that mechanics are critical to almost every single biological process we study. And while from the many talks we've seen today, we can determine the concentration of proteins, metabolites, nucleic acids, and in space and time in these cells, we know very little about the mechanics. And it's not only a fundamental question, it's also a question that pertains to disease states. So this is a plot from a company in Southern California called Foresight Technologies, and they plot various disease states, and they emphasize how different disease states have modified mechanics. And we saw examples of this from Maddie Parsons' talk yesterday, and there's many other examples of this throughout. Additionally, when I give talks to my colleagues in chemistry, I like to speak their language. And so one thing I like to emphasize is I say, look, if anybody wants to measure the concentration of a post-translational modification like the one shown on the left, you want to measure phosphorylation, you want to measure zinc, you want to measure glycosylation of a protein, you have catalogs of tools. In contrast, if you want to measure a conformational change like the one shown on the right for this alpha-2b beta-3 integrin receptor on the surface of platelets, you want to measure, quantify that mechanically driven conformational change, you're really out of luck. There's not many tools out there. And so that just underscores the need for the tools. So of course, this has been a long-standing problem. And there's a series of challenges that relate to mechanics. I have some of these listed here. It's primarily the issue is that forces, the molecular scale forces particularly, are just really small. They're at the scale of piconewtons. And if you prefer to use kts, so 4 piconewtons applied at a distance of a nanometer is a kt. If you prefer to use kcals per mole, it's 7 piconewton nanometers. That's roughly a kcal per mole. So they're small units of energy. Most of these are piconewton scale. Not only are they small, but they're oriented, they're transient, so they're brief, and they're spatially organized. And so they're hard to detect. And this is really the problem. And so we as a community have been trying to measure these forces for a long time. And so I'm going to give you a brief history of some of the tools available. And so if you start really, one of the oldest methods reported was in this 1980 Science paper. And so this is work by Harris, Wild, and Stopak. And they were wondering how much force or whether cells apply force at all when they're adhering or migrating on a surface. And so what they did is they made rubber substrates. And they cultured cells on these rubber substrates. And you could see from these images in this science paper, the substrates wrinkled. Okay? And this was a huge advance. It was the first confirmation that traction forces generated by cells are real, and they can be quantified. From then, of course, the field is grown rapidly. And I would say the gold standard technique that's used most often these days is traction force microscopy, often abbreviated as TFM. And so in TFM, you load fiducial markers into the polymer substrate, and you record the deformation of the polymer by measuring the dislocation of these particles. And so this is an example of a MDA-MB231 breast cancer cell and mapping these traction forces created by the cell. Again, very powerful, but often requiring computational analysis to reconstruct the force field that created this distortion. Complementary to TFM are methods that use micropillar arrays, or these sometimes are called needle arrays. They're typically comprised of PDMS, originally developed by Chris Chen and colleagues. And you essentially grow your cells on these bed of needles and record the deformation of the pillar. And from this, you can also infer the magnitude of the force. This is less computationally intensive because there's a spring constant for each one of these pillars. And from the distortion, you can determine the traction forces generated. OK, so this is a summary of some of the techniques out there. But I think many of us are interested in the molecular scale forces. And so to focus on the molecular scale forces, there's really three main families of approaches. And I have these highlighted here. And so on the far left, you have the genetically encoded tension probes. So these are typically a pair of fluorescent proteins flanking a extendable linker and inserted into the protein of interest you care about. The method in the middle, these are a family of methods that are single molecule in nature. These are very elegant, and they really underscore a lot of the techniques that and have been used to calibrate many of the techniques. I have AFM optical tweezers and magnetic tweezers, and we've seen some really beautiful work already in this meeting with AFM, for example. And then the technique I'm going to spend most of my time, all my time talking about today, are the molecular force sensors. These are outside the cell, anchored to a substrate, and used to measure the molecular forces transmitted by cell surface receptors. And I would say each of these methods has advantages and disadvantages. Okay? And if you're interested in talking about the other two methods, I'm happy to talk about them, and there's other experts in the field. We've contributed also to some of these other methods, but I'm not going to talk about them. We're going to dive directly into the molecular force sensors for today's talk in terms of technique. Okay, so let me show you essentially how these work. So essentially, it's a video that's going to run here, and it's a molecule that we immobilize on a surface. It's the two termini of this extendable molecule has a fluorophore quencher, also a ligand of interest, such that when a receptor from a cell surface receptor binds, nothing happens. But if there's a force, you stretch this extendable linker, and you generate a turn-on fluorescence response. And so what's nice about this approach is that as long as you know the relationship between the extension and the force, you can use the fluorescence intensity to infer the distance, and then from that infer the applied force generated between the receptor and the ligand. What I'm not showing, of course, here is that there's a cytoskeleton that couples to that receptor that generates that force that we're measuring. Okay, so when we started with these methods, we started with polyethylene glycol linkers because they're like entropic springs. And they essentially show functions like this. It's an analog function. So more force, more extension, and you can get a signal. Let me show you an example of this. And so this is going to be a video that runs of seeding a fibroblast on a surface. And so what you see in red, this is a GFP actin tag. And then what you see in green is our molecular tension probe. Again, it's polyethylene glycol. It's a 24-unit PEG. And you have a fluorophore at one end and a quencher at one end, and then a cyclic RGD peptide that engages the integrins. And you can see as the cell dynamically spreads, you see this green signal. And these are the integrins applying force and stretching these probes during adhesion and spreading. Okay, so we can use these. And what's really special about these probes is that you're using, in this case, conventional wide field fluorescence imaging. So this is compatible with all your other techniques. You can even fix and stain a cell. And so this panel of images here, we've fixed these cells. In some cases, you can do live imaging, as I've shown before. But you can measure things like the integrin location and its relationship with tension, paxillin, zixin, actin, all the components or some of the components of the focal adhesion complex. You could track those simultaneously while you track these mechanical forces. And so it offers you a way to study the relationship with force and protein composition. And in some cases, you could see interesting relationships. Like when you look at this beta-3 integrin and tension, you see a mismatch. The location of the integrin doesn't match perfectly with the location of the tension, meaning that the part of the focal adhesion matures and mounts tension after the arrival of these integrin receptors in this case. But we had, you could see here a heat map of force. We're estimating the force generated per integrin, and we saw units, average forces here that span up to 12 piconewtons. But this was problematic, and let me tell you what the problem is here. The problem is it's ensemble averaging. So each one of these pixels contains perhaps 10 to 100 sensors, and there's 10 to 100 integrins that bound. And so that while we know the absolute density per pixel, and we know the Fred signal in each pixel, it's still an average because the signal could be generated by one really active integrin or 100 very weakly active integrins. And so we don't know the actual force per receptor. And so to solve this problem, we thought we have to move away from an analog probe to a digital probe. And that's the next part of the slide I'm going to show you here. Obviously, the other solution was to do single molecule imaging. But in the early days, we're not very good at single molecule imaging. And so we didn't solve the problem that way. We solved it by switching to a digital probe. And so the probe we switched to is comprised of nucleic acids, DNA. And if you're interested in any of these tools or methods, really, nucleic acids are the best way to go. Let me show you why. This is because you can build fluorophore quencher systems that are brought together with a stem loop hairpin. And so the reason this is great is because your fluorophore quenchers start off with zero force very close together. So you get very low background. And then once the force is applied, you have a sudden, a switch-like response where you stretch the DNA and you get up to 100-fold enhancement in fluorescence. This is partly because it's not really FRET in this case, it's actually contact quenching, and so you really suppress the background. The other beautiful thing about these systems is that the stem here can be tuned. So you can tune that delta G of folding and then consequently change the F1/2, the equilibrium force that leads to unraveling of the hairpin and tuning of that response. So you can tune it from about 5 piconewtons all the way up to 20 piconewtons really easily. So let me show you an example of using this probe. I'm going to switch systems on you here. So this is now, you're going to see a video of a human platelet landing on a surface, and that's where the video is going to run. And so on the left-hand side, you see the reflection interference contrast microscopy. We've seen some IRM images thus far. This is time, this is a scale bar. You can see as the platelet spreads on the surface, you see a distinct bullseye pattern of tension generated by that platelet. This had never been seen before, and this exceeds the spatial resolution of traction force microscopy, so this was not visible previously. Okay, and the neat thing here is every time you see fluorescence, it indicates a 4.7 piconewton signal, and the signal intensity is linearly proportional to the number of integrins that are applying force. So we can quantify the number of receptors that are applying this 5 piconewton tug. But the question I think our collaborators were asking is, how much force is exactly being applied? Is it more than, all we know it's more than 5. So to answer that question, all we had to do is create a library of these probes where we tune the F1/2 from 5 to 13 to 19 piconewtons. And if you're following my talk, that means all we did is we increased the GC content in that stem. We went from 22% GC content to 100% GC content. And so now it takes about 20 kcals per mole for every receptor to open a hairpin, and now you only see very few receptors opening hairpins. So now we can define the magnitude of forces being generated by these platelets. The other really cool thing here, these are human platelets. And so we always joke in the lab that it's blood, sweat, and tears in the lab, and this is real student blood that goes into these experiments. Okay, so the next question we had, I'm going to walk through a set of questions, is are these really force probes? All we're measuring is the DNA unraveling. For us to say that this is really a force probe, we have to measure not just the magnitude, but also the direction, the vector of the force. And so to answer this, I'm going to highlight these challenges because they relate to microscopy, which I think is a topic of interest to everybody here. And the other reason that we wanted to measure force orientation is because there's papers out there. Again, I'm showing you the alpha 2b beta 3 receptor here. And this is a quote from Tim Springer. He says, "We believe that the application of lateral force is the most physiologically relevant mechanism of activating an integrin." So he's saying it's not pulling up that's important, it's the lateral force. And what we can see is just tension. We don't know. So to answer this question, we had this hypothesis, and this is beautiful work from Josh Brockman and Aaron Blanchard and Alexa Mathesis. And so what we did is we had this hypothesis that the DNA is like points toward the force. And so if the force is east, you get the DNA pointing that way. And so that was the first hypothesis. The other hypothesis is that we can use fluorescence polarization to determine that force orientation. Okay, and the lucky thing is that there had been earlier work by, in fact, Tajib Ha and colleagues showing from different techniques, both NMR and FRET, that cyanine dyes, they stack against the blunt end of a duplex. That means your organic dye, the cyanine dye, Cy3 or Cy5, is aligned to the long axis of the duplex. And so what we assumed is that that means the transition dipole moment of this organic dye points toward, helps us identify the location of the force. So again, if this is a duplex and the force is this way or the force is this way, the transition dipole moment can be plotted like a cylinder that points in each of these directions. And so this is all we thought of the DNA as really a cylinder with an orthogonal transition dipole moment to that. Okay, so what we had to do is do polarization imaging, and so we did the poor man's version of a polarization imaging, and this is excitation resolved fluorescence polarization, which means we just put a half wave plate in front of the laser, and we rotate it, and we just measure the intensity on our camera. So if the laser is aligned with our dye, you get very bright excitation, and if it's orthogonal, you get very dim. Okay, so let me show you what this means. And so this is a human platelet that's sitting on a surface, and this is the tension map. This is what you see in terms of hairpin opening. But then if you take a look at this 1 pixel, we start with an angle of 0 degrees, and we shift the half wave plate all the way around, and we measure the intensity. It goes bright, dim, and bright. Okay, so the amplitude here gives us the tilt of the force, and the phase gives us the orientation of the force. And again, if it doesn't make sense to you, just imagine a duplex pointing straight up. If I go S or P polarized, it's going to be identical. It'll be a flat line. Okay? But as soon as I get more and more tilt, you get more and more amplitude in this function. So the amplitude gives you the tilt of that cylinder. Okay, so we did this, and we applied it to our platelets, and now our image is lit up such that we can determine a bar indicating the ensemble. There's an ensemble for approximately 10 to 50 receptors in each pixel, but it tells us the tilt. So the color indicates tilt, and what Tim Springer said may be true, but all we know is that there's a significant portion of these receptors that are applying very tilted forces. And in fact, if you look at these bars, they point toward a pole at the center of the platelet. So platelets do apply forces contractile that are pointing inward toward the center of the platelet. And we think this has a role in the function of platelet and clotting functions. Okay, so the next tool I'm going to show you focuses on super resolution maps. And the question that we had is, how can we see these forces at super resolved scales? And so again, this is our team that worked on this. And we all saw Ralph Youngman's talk a few days earlier. And so I had seen some of Ralph's talks, and I thought, oh, this is going to be so easy. So I brought the graduate students in the lab. I said, very easy. So Ralph, all they do is they need DNA to link to bind and unbind reversibly to a docking site. And we already use DNA. So we thought, oh, this is going to be very easy. All we have to do is have our DNA probe, and then at the site, when it opens, this becomes a docking site for the imager. And so Josh and Han and Aaron, they go to the lab, and they spent approximately 6 months, and they get very bad data. Regardless of what we try to do, we get very few localizations. So these images here are collected with this fluorophore, but if we try to get the docking strand to bind here, it doesn't work. So this really, it doesn't work very well. We weren't smart enough to recognize what we learned later, is that when you stretch a piece of DNA and you apply 20, 30 piconewtons of force on a piece of DNA, it creates a barrier to hybridization. Okay, so just imagine stretching a piece of DNA. It doesn't want to coil to make a B-form DNA duplex. And so what we have to do is redesign the structure, as shown here. So essentially, we have a stem here, but it doesn't have a loop, such that when force is applied through the blue-black duplex, it's under strain. But then once it dissociates mechanically, this blue strand is now force-free. The force gets funneled or transmitted to the green oligo, and it leaves the blue one behind. So the blue one is relaxed now. And now when it's relaxed, you can get reversible binding and unbinding, and you get nice localizations. And so once we did this, this is the raw data, so just so you could see. This is time. Again, these are human platelets. You can see the blinking underneath the platelets. We do the classic reconstruction, and now you can get these force maps at very high spatial resolution. This is one platelet that formed filopodial spiky structures, and so this is why I'm highlighting it, but many of them also make those structures that are bullseye shaped. And you can see now we can zoom to resolutions close to the resolution of integrins and integrin assemblies. But again, I want to remind you, we're not looking at integrins, we're looking at maps of the biophysical mechanical events that are generated by the integrins in this case. Okay, so this is great. I'm going to continue telling you about tools and questions. And then the next question that we had that started a few years ago really had to do with the rate of pulling. Okay, so if you look at the literature, there's quotes, and I'm highlighting this quote from this nice paper from Pereira-Roca-Cusax. And in this paper, they say the range of loading rates, this is how fast you pull, it's hard to assess. But they guess that the forced loading rates could range from 10 to 10 to the 5 piconewtons per second, okay? So if you just imagine the range of expected force loading rates, it's massive, right? And the problem is nobody really knows. And the other problem is that loading rate dictates the response to force. So if you pull really fast, you get one type of unfolding biophysical response versus a very slow response. Even the probability of unbinding depends on the loading rate. Okay, so we set about to try to answer this question. And our solution to the problem is really, it's illustrated with this cartoon. So if you imagine two light switches in series, and you move your hand at a certain speed, where each light bulb gives you a different color, you would time yellow, red, and the time delay between yellow and red tells you how fast your arm's moving. So this is, I want you to imagine this situation. And this is all we did here, okay? And so what we had to do is design a probe that produced two colors, okay, at each different force magnitude. And so this is the design. It had an oligo that had two quenchers, a donor, and an acceptor. And the idea is that when it's relaxed, there's no signal. It's quenched. Under the application of force of about 5 piconewtons, which is the force to unravel the stem loop hairpin, you separate the donor from the quencher so you get a green signature, green flare. And then in some later time, you can shear that double quenched oligo, and then you get sensitized fret signal. So you go donor, sensitized fret, and the time delay is the loading rate. This is the type of signal we see and we expect it to see. Again, this is 5 piconewton force, and then the donor gets dim and the fret goes up, and that's 50 piconewton force. So this is the delta T, allowing us to estimate a linear loading rate for how fast an integrin is pulling. So we do this. This video is going to just run you through what the data looks like. And so this is RICM overlaid with a single molecule traces. So this is each molecule. This is what the data looks like. I like to show with the raw data because sometimes it's hard to imagine what these experiments look like. So these are each event, and then you could see we see green followed by red. These are the ones that we saw complete shearing. There's many more events where we see green that lights up and dies, but then a few of them have green followed by red, and these are the events that are being highlighted in this video. In any case, we analyze thousands of events, and we get a conclusion that says that we conclude that the loading rate is 1.1 piconewtons per second. And we published this in JACS. What was interesting is that many other teams were actually doing this simultaneously alongside our work. And so TJ Ha earlier last year published a paper in Science that showed that in fibroblasts, the same cell line that we used, they reported a loading rate of 0.5 to 4 piconewtons per second. And then my former postdoc, Zheng Liu, who has a lab at Wuhan now, also did the same measurement, and they report a 0.5 to 2 piconewton per second loading rate, again, using integrins in fibroblasts. And so this seems to be a universal feature of living cells, that they tend to generate forces at these time scales within active focal adhesions. Okay, so this is, I think, something that's developing in the field. Okay, so the next question, and this is, I get this a lot when I give talks. People say, well, hold on, you're putting this probe on a glass slide. This is a very stiff surface. It's very different than having a surface that's ECM or that's softer. So how do we know if this is true or if this is an artifact of the surface you're looking at? And so I'm going to show you some techniques where we immobilize these probes on fluid membranes, okay? And so one approach, which was really driven by a talented grad student, Yusong, what Yusong did is he created a DNA origami platform. He called it the DNA origami tension sensor. the dot, or the dots. And so in the dots, the bottom of the origami structure has a series of anchor strands so that it can anchor to a membrane or anchor to a cell. And then the other face is the DNA tension sensor, the hairpin probe that I just showed you. And this is the design from this is AFM imaging of these structures. They're approximately 80 by 40 nanometers in size. And the key thing is that we separate the force sensor from another force sensor by distances that often exceed 100 nanometers, so we don't see intermolecular fret, so that we can have a nice measurement on fluid interfaces. Additionally, we put a set of organic dyes on the origami structure so we can measure the density of the dots probe. So we know the density and we know where the force is being applied. Okay, so this is going to be a video. I'm going to switch the biology on you again. And so what you're going to see here now are these dots probes with peptide MHCs. These are antigens specific to the T cell receptor. And you're going to see a TCR, a T cell receptor that lands on a surface, and that's the video that's going to run. So first you're going to watch these T cells land, and they create a dark spot. Then it's followed by a bright accumulation of signal in the center. So the first reason that you see the dark spot, this is actually the compression pushing force of the T cell. So the T cell pushes and crowds out and excludes these probes initially. But then later on, the TCRs bind their antigen, and then they pull them and accumulate them in the center of the junction. Okay, and so once we quantify the signal in the middle, this is what this next slide shows. It shows quantification of these forces. And this is a time-lapse video, and you can see the tension. So the tension is really a ratiometric measurement of the density versus the hairpin opening. And from the ratiometric imaging map, we can determine the force that's being applied here. And you can see that, indeed, these forces do exist even on fluid membrane interfaces. Additionally, you can take these dots probes and anchor them to another living cell. And in this case, we took a B cell. And then what we did is we brought the B cell in contact with the T cell. And at the junction between the two, you can use confocal imaging and image the junction. Again, you see the hairpin opening, you see the probe accumulating, and the ratio gives you the tension. So you see maps, hot zones of mechanical force at live cell-cell junctions in this case. So these forces are real. They exist, particularly in this case for the TCR, which it's a different story here, and it's very important and relevant to the activation of the T cell receptor. Okay. This is going to be the final part of my talk. And this is a challenge, I think, to our lab and to many in the field. And that is, how do we democratize these probes outside of really specialized labs and outside of the use of a 100x oil objective? Okay, so a lot of times we go to collaborate with colleagues and they say, oh, but this 100x oil objective with turf imaging, this is very hard for us. We can't use this. So how do we solve this problem? And so I'm going to show you a set of techniques, what I think will help us democratize this technique and make the probes really available to the wide cell biology community. Okay, so the first technique I'm going to show you allows you to use these probes to analyze force using a flow cytometry methods. Okay, and the technique is shown here in this slide. So essentially what we did is we created a different design for the probe. Shown in the left is essentially what we created, which is a blue strand that's anchored at one end and has the RG peptide on the other end. And this probe is now covalently linked to the slide, so that when the force is applied to it, it really just destabilizes the duplex, the red oligo. So as you pull on it more, the B-form DNA gets unstable, the red comes off, and now you have a released oligonucleotide. And what we did is we introduced a cholesterol unit on the oligo such that this now stains or tags the cell that generated the force. And so once the cell is tagged, you can scrape it or collect it off the slide and run it on a flow cytometer, and flow then gives you a barcode of tension. Okay, to convince ourselves that this works, we did some simulation of that type of pulling. It's called 3', 5' pulling on the blue oligo and watching the red one come off. And you'll see the video as the force ramps up, the complementary oligo falls off. And we saw that it takes approximately 40 piconewtons of force to drive that dehybridization event. Okay, so to convince ourselves that this works in cells, we took our same cells and we tagged them with a donor and an acceptor. It's not a dark quencher anymore in this case. So you can see the Cy3b, the donor, gets bright underneath this fibroblast, and the acceptor gets dark because the acceptor is now lost. And in fact, it's lost and stuck to the cell membrane in this case. You can see these maps. Donor brighter, so that means your acceptor gets darker because you lose the fret. And so in the bottom of the slide, I show you a set of experiments and controls where we had our TAG probe. This is our tagging probe. And then we had a system where we withheld the cholesterol, or we withheld the RGD peptide, or we added latrunculin, which inhibits actin polymerization. And then when you analyze these experiments, you could see only when you have all the components, the cholesterol, the RGD, and the cytoskeleton intact, do you see effective tagging of the cells. And then if you, all the experiments where you withhold these, you lose the tagging. You get very low background level tagging. And so this opens the door to using flow cytometry, which many folks in the cell biology community like to use rather than microscopy. Okay, another technique that would be powerful, again, along the same line of democratization, is to integrate mechanobiology with omics. Okay, we saw spatial transcriptomics, spatial proteomic studies in the last few days in the seminars, and we want to introduce mechano-omics to this field. Okay, so that requires creating the right kind of probes for this technique. And so there's many ways to do this, actually, but this is one way we envisioned and we implemented recently. And so the idea was to combine the DNA tension probe with a field called proximity labeling. So in proximity labeling, what you typically do is you add an enzyme to the protein of interest, and the enzyme generates a reactive species that covalently tags all the proteins within a neighborhood of the protein of interest. And the most famous examples are BioID or TurboID or Apex2. These are many common techniques to do proximity labeling. Alice Ting is one of the pioneers in this field, and we basically took the same protocols that she and others had developed. And so what we do now is it's the same system. The hairpin opens, and then what we do in this case is that we bring in a complementary ligo, but rather than have it tagged for paint, in this case, it's very stable, it binds, and it brings with it an enzyme, and this is horseradish peroxidase, that in the presence of hydrogen peroxide and biotin phenol generates reactive species that covalently tag all the proximal proteome. And in this case, we're going to focus on the T-cell receptor because the partners of the TCR really were unknown in terms of the mechanical partners of the TCR during mechanotransduction. So we ran some microscopy experiments initially to confirm that this works. And this is what you see here. So you essentially, we add biotin phenol and peroxide for 1 minute. And you could see this is the Cy3b opening. And this is streptavidin staining the mechanome that's nearby the TCR. And you could see similar patterns. If we don't add the peroxide, but add biotin phenol, you get no streptavidin staining. And this is what you see in the control. Okay, so the microscopy works, and we're not surprised. But we next had to get this to work for proteomics. This is what the workflow looks like. Okay, we had never done proteomics before in our lab, and so this was new to us. So what you have to do is essentially run the same experiment, and then scrape up all your cells, run them on a streptavidin pull down all the proteins that are biotin-lated, and then send them out to mass spec. The first problem that we discovered is you need larger quantities of protein. And so what we had to do is scale up our microscope slides. So we set up these 1 by 3 inch full glass slides that we load with about 10 million cells in each slide. We load them in, and we run the same protocol that I just described to you. Okay. After we load them in, we scrape all these cells, we pull down all the streptavidin, and then we run proteomics. And this is, I'm going to share with you here, this is the first mechanomic trial or experiment where we're mapping out the proteins that are recruited in proximity to mechanical forces. Okay, and what's really neat is many of the proteins we identify are many of the co-receptors and the proteins involved in the TCR complex, in the CD3 complex. And so we identify proteins like CD3 epsilon, delta, the alpha and beta subunit, gamma, and as well as the co-receptor CD8, and many of the other partners in the signaling pathway. We also identified some proteins that had not been seen before, and we're investigating them now. Many of them actually are involved in adhesions, and so many of these undiscovered receptors are involved in T-cell adhesion. We think they contribute to the TCR mechanotransduction. Okay, this is great. So this is going to be the last part of the talk, and this is the last part of this democratization effort that we've been running. And so we thought about it some more. We thought, really, catalytic amplification is a cornerstone of modern biochemistry. And so if you look at modern biochemistry, it's really the PCR and ELISA reactions that underpin much of what we do. And so we thought if we can create PCR-like reactions for mechanics, this would make it much easier, much more convenient to map forces. And so what we did is created an assay that uses a polymerase type reaction. Okay, the reaction that we ended up using is driven by an enzyme called Cas12a. It's a cousin of Cas9. The difference is Cas12a can be designed to activate in response to binding single-stranded DNA. So essentially, the surface in this case was double-stranded DNA, and it was a monolayer of double-stranded DNA. That the guide RNA is unable to bind to because it's duplexed. But when there's a force, you mechanically denature the duplex, exposing this hidden cryptic site, which then recruits the guide RNA with the Cas12a. Once it binds, it unleashes something called transactivity. Transactivity means it destroys all single-stranded DNA it finds in solution. And we load the solution, the 96-well plate in this case, with fluorogenic nucleic acids. And so suddenly there's a massive boost in signal, and it allows us to detect the mechanical forces using a plate reader, a fluorescence plate reader. The limit of detection here, turns out it's approximately 2,000 molecules, 2,000 single-stranded DNA in a 96-well plate. What's interesting, the limit of detection is not in molar units now, it's in units of molecules per unit area because it's a surface immobilized template for amplification. In any case, we run the assay, and I'm going to show you examples of running this assay using human platelets again. You don't need many, so a small volume of lead, a few microliters, has plenty of platelets to get signal from a single 96-well plate. And so you don't need a fancy microscopy. I know this is sacrilegious in a meeting like this when I say you don't need microscopes anymore. But it's just convenient to run. And the nice thing about running it for platelets is these are primary samples from a human, and they're independent of platelet count, which is a problem that's encountered typically in these assays. So here's an example of how this is powerful. You could draw a single blood draw as a few milliliters of blood, and that allows you to do thousands of measurements, of mechanical measurements of platelets. And so what we did is we aliquoted that blood draw, those platelets, in 96-well plates, and then treated each well, or triplicates of wells, if you will, with a different dose of anti-platelet medications. Okay? And now we can determine a Mechano IC50 for a pharmacological agent. Okay? And what's neat about this is all data from a single human, from a single graduate student donor. And so you can determine the sensitivity of an individual to aspirin, to various other inhibitors, and not every individual is a responder to aspirin. I tested myself, for example. I'm sensitive to aspirin, but not everybody is. At least their platelets aren't. And so this becomes a way to get personalized determination of activity of an individual's platelets. This led us to, this is going to be the last story I'm going to share with you. So this led us to using this assay to detect a clinically relevant disease, which is called heparin-induced thrombocytopenia. It's a rare condition where essentially a patient develops antibodies against a very commonly used blood thinner called heparin. So everybody uses heparin when you do surgery. Surgeons love it. But something like 0.1% of patients develop antibodies that essentially drive thrombosis and clotting, and in some rare cases, they lead to thrombotic events, amputation, and death. So it's really dangerous. And it's very important to test frequently for this condition. The current standard test for this is either an ELISA, which turns out to be really has very poor specificity because there's many antibodies and only a subset of them are pathogenic. The other technique is a functional assay that uses a radio-labeled isotope, which is very difficult to run. And so we employed the assay that I just shared with you, and we actually launched a clinical trial. This is the first human clinical trial for using a molecular mechanobiology assay. We recruited patients, and then we ran the standard of care diagnostic, and then we ran the assay in our lab at Emory. So the student would walk over to the hospital, pick up the blood, and come back. We separately ran the two assays. We're blinded to everything, and so far we see 100% agreement between the standard assay, which is a functional radio-labeled isotope assay, and this Mechano assay that I just showed you here. Okay, I'm gonna end my talk rather than concluding, just ending with questions. I find that, you know, many talks, you see the talk and you think everything's been answered and we have all the answers, and really it's just the tip of the iceberg. There's so much we don't know, and I just want to set the stage and tell you how little we know. And so, for example, we don't know the relationship between the tension and the receptor conformation. So these integrins have conformational changes, the TCRs have conformational changes. We still don't know the relationship between force and conformation. We don't know the relationship between the duration of the binding event and the duration of the force. If we can measure these things, we can determine if there's such a thing as a catch bond, if the bonds stabilize with force. Because these are measured with single molecule techniques, but they've never been recorded in a living cell. What's the peak force? we still don't know this. We assume the loading rate is linear, but what is the actual loading rate? We don't really know. We see a lot of dynamics. What drives the force dynamics? What are the adapter proteins that are driving these forces? And then there's over 20 different alpha-beta integrins. We don't really know which ones are transmitting the forces that we measure. There could be different subtypes that have different magnitudes of the force, and we don't know the answer to this. And then the final thing I want to leave you with is I reported everything in units of force, but it's really energy, because it's a distance that the molecule has been extended. And we don't really know still. Should we be thinking of these in terms of energy? Should we be reporting kcals per mole or kTs? Or should we be thinking about these events as being important? Is the number of piconewtons important, or is the joules important? And we don't even have the answer to that question. And so there's still a lot of mysteries here. Okay, a few advertisements before I conclude and stop here. So we have an NIH-sponsored center that's focused on molecular mechanobiology. If you're interested, we have seminars every month. So if you scan the QR code, we can add you to the sign-up list, and you can get emails about these seminars. We have weekly office hours, so if you want to use these probes, we're happy to give you protocols. We also share reagents for free. So if you want any of these probes, just contact us. We'll ship them to you. We do, like I said, free consulting. And we also, every year, we have a workshop, a miniature, very, very mini version of this meeting. And so you do labs for 4 days, and we have talks. And so please reach out to us if you're interested in collaborating. Additionally, we have a Gordon Research Conference that I'm chairing in this summer. That's on biointerfaces. So it's molecular design, characterization, and manipulation of living, non-living interfaces. It's going to be this summer in Italy. And so if you're interested, please apply. We'd love to see you. There's going to be some mechanobiology and microscopy highlighted in this meeting. Lastly, I'm going to say we're hiring postdocs. This is a photo of the building we're in at Emory. This is a nice picture of Atlanta in the fall when the leaves change color. And this is downtown at Atlanta. And so I'm just going to end. This is a photo of the lab. And then here's acknowledgments. And I thank you again for your attention.
Yeah, thank you, Khalid. Have you tried to measure the loading rate on these fluid substrates or on soft substrates? And related to this, what's the time scale that you measure this loading rate event? And what's the time resolution that you can get?
Yes, great question. So the first part of the question asked about, have we measured this loading rate not on a hard gigapascal hard slide, but on a fluid membrane or elastic substrate? And the answer is no. We have not done that. The problem with trying to do it on a fluid membrane is things move laterally, and so it becomes more challenging. I suspect One of the teams that are doing this, so either TJ or Zheng Lu's lab, will probably report this in the coming year. But it's a really interesting question because the force magnitudes are really suppressed on soft surfaces. And so it's not clear, would the loading rate also be suppressed, or is it just the force magnitude that's suppressed on the soft surface? So it's a great question. In terms of our time resolution, it's the time resolution of doing imaging with an EMCCD. So we're typically taking an image every 100 milliseconds or 200 milliseconds. We can't really push it much further than this with our capabilities, with the brightness of these dyes. That's our time resolution. So yeah, well, just like that video I showed you, we'll just watch a cell for 5 or 10 minutes, and we'll track these events across that time scale. So the surface has a density of approximately 500 ligands per square micron. Only 1 in 1,000 of those are fluorescently labeled. So it's a sparse density of reporters so that we can do single molecule. That's typically how we run these experiments. And we've only done it with cyclic RGD. And interestingly, the other 2 teams also did it with cyclic RGD. So even the loading rate may change with a different fibronectin or an RGD. Yeah.
Over here. Thanks a lot. Very interesting. I'm wondering, any ways to use your probes intracellularly, maybe microinjecting them and targeting them to specific places?
Yeah. There is one report of another team that has microinjected the DNA attention probes inside cells. But I would say if you're interested in forces inside the cell, you really have to use the genetically encoded tension probes. And there's different ways of using these probes, but that's the preferable way to do it.
But why? Because it seems like your approach is much more tunable. You could target things a bit more precisely. Some things are not really easy to genetically encode.
No, I think the team that microinjected the hairpin probes, they saw some nice signal, but the 2 termini of the DNA have to bind 2 different elements inside the cell. And so they had an actin binding domain and another domain that binds another component in the cell. And it will accumulate in different sites of the cell, and you can image. So you have to find appropriate ligands if you want to do this. But yes, it's certainly doable. I still think the genetically encoded probes, because it could allow you to do things in a living organism, in an organoid, where microinjection is difficult to use. This is where, but certainly there's a place for each type of technique.
Thanks.
Hi, over here. Thank you very much for the nice talk. I was just thinking, if you were measuring within the cell, would you expect, because the DNA is, the backbone has got a lot of negative charges on it, would in different parts of the cell, if you've got different salt concentrations, how would that affect, the charge shielding on the DNA? Does that change the force that you read out?
Yeah. So the question is, does the ionic strength or the salt concentration change the readout? Oh, certainly. And so I'll give you an example of this. And so when we did the paint imaging, we used Ralph Youngman's fast paint approach, which used 75 millimolar magnesium concentration, 10-fold or greater than the usual magnesium concentration. And we had to adjust all our calculations because the delta G of hybridization is greater and the F1/2 is increased with more magnesium. Most of these probes are outside the cell, and we assume a constant divalent cation and monovalent cation concentration, and so we just use the buffer ionic strength to estimate the force. We don't expect to see huge changes, but if you do add more magnesium or sodium to your buffer, you will shift that F1/2. And if you look at our website for the center, we actually have a simple online calculator. You could put in your DNA sequence or the delta G of hybridization, and it will give you the F1/2. So there's a convenient way you can calculate it very easily if you'd like.